The trip operon in bacteria encodes enzymes needed for biosynthesis of the amino acid tryptophan. The trip operon is regulated both by a repressor and by attenuation. This animation will present a model for how attenuation works. Overall, attenuation in the trip operon can prevent expression of tryptophan biosynthesis genes when tryptophan levels in the cell are high, essentially turning off the trip operon when it isn't needed. Attenuation of the trip operon involves structures and sequences of the TrpL mRNA. Interactions among complementary sequences of the TrpL mRNA can form stem loop structures in the RNA transcript. 4 regions are involved. These are numbered relative to the 5' end of the transcript, with 1 closest to the 5' end. Region 2 is complementary to both regions 1 and 3, and can form a stem loop structure with either of them, but not with both at the same time. Similarly, region 3 is complementary to both regions 2 and 4. When regions 1 and 2 form a stem loop, region 3 forms a stem loop with region 4. Immediately following region 4 is a uracil-rich sequence called the attenuator. When the region 3-4 stem loop is present, it acts with the attenuator to form a terminator for transcription at the end of the TrpL gene. When the region 2-3 stem loop is present, the terminator structure does not form, and transcription proceeds to the TrpE gene and other tryptophan biosynthesis genes in the operon. There are 3 scenarios possible with respect to attenuation in the trip operon. If transcription is occurring alone in the absence of translation, then the TrpL mRNA will take on its most stable conformation. In that form, the region 1-2 and region 3-4 stem loops form, and transcription is terminated at the end of the TrpL gene. The other 2 scenarios involve translation coupled with transcription. The difference between these 2 scenarios depends on whether or not the ribosome gets stalled in the region 1 area. 2 adjacent tryptophan codons in region 1 determine whether or not stalling will take place there. If tryptophan levels in the cell are low, then tryptophan transfer RNA levels will also be low. This will cause the ribosome to pause at the 2 tryptophan codons as it waits for tryptophan transfer RNA. The location of the ribosome at this pause will shield region 1 from region 2, thus freeing region 2 to form a stem loop with region 3. Since this prevents the formation of the region 3-4 stem loop, transcription is allowed to proceed into the tryptophan biosynthesis gene in the operon. If tryptophan levels in the cell are high, then the ribosome will not get stalled at the tryptophan codons in region 1. The ribosome will continue until it reaches the stop codon for the TrpL gene, where it will experience the normal pause that occurs during translational termination. While paused at the stop codon, the ribosome will physically block region 2 from interacting with region 3. Thus, the region 3-4 stem loop will readily form, leading to termination of transcription before the tryptophan biosynthesis genes are reached. In this way, attenuation turns off the trip operon when tryptophan levels are high.